long time ago, in cells named eukaryote and prokaryote, lived two races of tubulin subunits, alpha and beta. They were both descendants of the globular protein race of tubulin, and they both contained GTP in their structures, which they used to attach to other subunits. But they had one distinct difference. Alpha subunits never hydrolyzed their GTP, while beta subunits always hydrolyzed their GTP to GDP during attachment. Unfortunately for both races, they could never attach to someone of their own race. For a while now, the tubulin subunits noticed that the cell around them was falling apart at the seams. The walls were collapsing without structure, and the organelles would just float around without direction. The motor proteins could no longer transport cargo from one area to another, and their cells could no longer complete division. One day, alpha and beta were both in animal eukaryote bar, mourning the end of their existences with copious amounts of alcohol while studiously ignoring each other, when they came across someone named centrosome. Centrosome was composed of two centrioles at right angles to each other, surrounded by paracentriolar material. The centrioles were arrangements of 9 microtubule triplets in circles, while his paracentriolar material was made up of proteins. Centrosome told them of a time when their two races got along, and their united work caused the cells to be stable and at peace. Inspired by Centrosome's talk, Alpha and Beta asked if he could teach them to work together to save their cells. Centrosome said he would. He could do this because Centrosome had earned the title of MTOC Master. Microtubule Organizing Center Master, that is. Centrosome taught alpha and beta to use the force to unite themselves together into one full tubulin protein. He said that since they could not bind to those of the same race, they would have to bind to each other using non-covalent bonds in an alternating pattern of alpha and beta. Alpha would bind to beta using his GTP without hydrolyzing, and beta would bind to another alpha by hydrolyzing his own GTP to GDP. This would continue longitudinally until a long protofilament was formed. To keep things organized, alpha was assigned the negative end, while beta was assigned the positive. To make themselves stronger, centrosome taught them to bind laterally as well. Only in this way, their protofilaments would line up negative ends together and positive ends together, but their non-covalent bonds formed alpha to alpha and beta to beta, without the use of GTP. Finally, once 13 of these protofilaments were bound together in a ring, a microtubule was formed. Gamma-tubulin, centrosome's previous students, who were also descendants of the globular protein race of tubulin, would then lead them and cap off their filament. Centrosome said that they were as strong as he could make them, but to beware of colchicine, because it can tear them apart again. Once assembled into teams of microtubules, they were able to complete their mission to save their cells. Many went out to keep their cell walls and membranes from collapsing in on them, and to hold their cells' organelles in place. Others went out to make roads for motor proteins to move along for transport, while others went out to form the ever-important spindle fibers used in eukaryote cell division. Some teams completely left centrosome, while others stayed in touch, and many took on different patterns in different cells. But it is thanks to all of them, with the help of centrosome, that the cells were saved and their lives were once again peaceful.